Hi everyone, welcome back to a new video. Now, this is a special lecture that gives an introduction and overview of biological agents. Now, do you know that between 2022 and 2024, about one third to half of all approved new drugs by the US FDA were biological agents? The increasing portion of biological agents in the FDA approvals suggest a growing trend in biopharmaceutical innovation. So, what are biological agents? Now, in this lecture, we will define biological agents, describe the general principles of biological agent production, and compare and contrast the pharmacological differences between small molecule and biological drugs, and discuss the hurdles of biological agents. So, biological agents these days are used for a wide range of diseases and conditions, including some very serious or life threatening cases such as cancer and rheumatoid arthritis. And here are the examples of biological products. First, we have Botox, a bacterial toxin that has dermatological and neurological uses. Now, the second one here is Herceptin, which is a mAb, a monoclonal antibody specifically for the HER2 positive breast cancer. And the third one is vaccines. Now, vaccines, for example, shingle vaccines, flu vaccines, and COVID 19 vaccines, they are all under the FDA's definition of biological products. And the last one is Embryo, a monoclonal antibody indicated for rheumatoid arthritis and psoriasis. So, in other words, biological products or biologics are medical products, aka drugs. Now, it could be made of sugars, proteins, nucleic acids, or any combination of these, or maybe even living entities such as cells and tissue. And like other normal small molecule drugs that we are used to, biological products are used for either treatment or cure of disease or medical conditions, prevention and diagnosis of diseases. These biological products have a common characteristic, which are made from a variety of natural sources. Now, we can use mammalian cell cultures to make biological products such as CHO cells or Chinese hamster ovary cells can be manipulated to produce the proteins of interest. And those proteins could be human proteins such as human cell receptors or physiological active proteins. And the bacteria, we can also use it to do the same job. For example, I use bacteria to make Taq DNA polymerase, but someone can also use bacteria to make many other proteins. One of the protein based COVID 19 vaccines made by Novavax used insect cells to generate the spike protein, so that's another great example. Traditional flu vaccines were cultured in eggs, so they have a warning for people that have egg allergy. Plant cell culture can also be a source material, and yeast can be very, very useful. Now, we will discuss yeast technology and how it can help us produce human or humanized antibodies that can be useful for treating certain diseases in the Biotechnology Series. And what are humans doing here? Now, yes, humans are also a source of biological material. Some of the newer treatments actually use cells as therapy. The company can harvest a patient cell and then ship that into the lab and do some manipulations or reprogramming, and finally infuse the same cell back into the patient to treat certain diseases. So, we touched upon the types of biological products at the beginning, which could be cells, proteins, human tissues, and vaccines. And depending on the type of vaccines, it could be inactivated viruses, mRNA, and protein. And there are also many different proteins, including small peptide hormones such as insulin or large proteins such as monoclonal antibodies. Cellular or gene therapy is also a hot topic that is gaining much interest. And now let's look at the major differences between small molecule drugs and biological products. The first is size or weight. Small molecule drugs tend to have a low molecular weight. They typically are in the range of a few hundred grams per mole. And in contrast, biological products generally have a high molecular weight and often measured in kilodaltons. Now, this means we are dealing with large, complex proteins when it comes to biologics. Small molecules, I should say, are typically synthesized through synthetic or chemical processes, and where biological products are produced using living cells or organisms. Because biologics rely on cell based production, there is an inherent risk of contamination. Additionally, small molecule drugs generally require fewer critical manufacturing steps while biologics involve many intricate steps to ensure consistency and safety. When it comes to structural characterization, small molecules are well defined. We know their exact chemical structure, and in contrast, biological products are more complex and may not be fully characterized, and their structures can vary. And for highly intricate biologics, we may not even have a complete understanding of their precise molecular composition. Another key difference is homogeneity. Small molecule drugs are highly uniform even if they exist as RNS enantiomers or racemic mixtures. They generally have the same pharmacological effect. Biological products, on the other hand, are heterogeneous, meaning they may contain variants. And in biologics industry, we refer this as batch to batch variation, which occurs because each production run depends on living cells. Now, once a batch is produced, the cells used will eventually die, requiring a new set of cells for the next batch, introducing inherent variability between batches. Immunogenicity is another major distinction. Small molecules are generally not immunogenic, although some exceptions exist, such as penicillin allergies. However, biological products, being foreign proteins, are inherently immunogenic, meaning they can trigger immune responses. And to mitigate this, biologics often require chemical or surface modification to reduce their immunogenicity. Now, this concept ties back to what we learned in immunology, highlighting the immune system's recognition of foreign proteins. Additionally, there are significant differences in the mechanisms of action, pharmacokinetics or PK, and pharmacodynamics or PD. Now, pharmacokinetics refers to what the body does to the drug, while pharmacodynamics describes what the drug does to the body. For small molecules, PK and PD studies are generally less complicated and more straightforward, and their analysis can often be conducted using in vitro techniques. However, for large molecules, such as these biologics, PK studies are significantly more complex. Moreover, when accessing these pharmacodynamics, that the involvement of the immune system adds another layer of complexity, making the study of biologics more challenging than that of small molecules. Now let's use a simple schematic to illustrate how biotechnology is used to produce biological products. Today, there are many different production methods, but this represents one of the more common or traditional approaches. First, living cells such as bacterial, insect, plant, mammalian or E cells are cultivated in a broth under specific growth conditions, and these conditions stimulate the cells to produce this desired protein. Now, I've personally worked on these processes in the past. During my time in school, I worked on producing antibody fragments using yeast cells. These E cells required precise temperature control to start secreting the antibody fragments. In essence, the process involves growing cells in a controlled environment where they generate the target protein. Now, once the protein is produced, it must be harvested from the crude mixture. This typically involves column chromatography, which separates the protein of interest based on size and charge differences from other unwanted proteins. The extracted protein is then concentrated, often measured in units per milliliter. Now, finally, the purified protein undergoes formulation and, if necessary, surface modifications to enhance its stability and bioavailability, transforming it into a therapeutically active drug. The biological products face several common challenges due to their complex production process. Unlike small molecule drugs, their manufacturing requires tightly controlled conditions to ensure product consistency, safety, and potency. One major challenge is contamination control, since biological products are derived from living cells. Their production environment must be carefully regulated to prevent contamination. Temperature and light sensitivity are also critical factors. For example, mRNA vaccines such as COVID-19 vaccines require ultra-low temperatures for storage because RNA is highly fragile and degrades easily. In contrast, protein-based biologics typically require refrigeration, which is easier to manage but still more demanding than small molecule drugs, which can often be stored at room temperature. The insulin and other biologics must be refrigerated, whereas most small molecule drugs can simply be stored on a shelf. Additionally, shelf life limitation pose another challenge. Now, while preservatives can sometimes extend the stability of biological products, they may also introduce uh, allergenic risks or affect product integrity. Now, this makes formulation and storage conditions even more critical in biological drug development. Since monoclonal antibodies are the most common biological drugs, let's have a quick review of monoclonal antibodies and its production. So, what is it? Monoclonal antibodies are identical copies of one type of antibody, and they are formed by fusing a spleen cell with a cancerous white blood cell. Now, the process begins with a mouse being given a vaccine with an antigen in it. Now, this triggers the formation of B cells, which make specific antibodies to match the antigen. And the mouse undergoes an operation and spleen cells, which forms the B cells, and in turn, the antibodies are collected. And the spleen cells are fused with myeloma cells and forming this hybridoma cells, and these hybridoma cells can divide indefinitely. The antibodies are then collected because these hybridomas can make a lot of antibodies in the process. Now, the uses of monoclonal antibodies are pretty wide. They can bind to specific antigens on cancer cells, and they can also come together, which makes detecting the cancer easier. And they can also support in carrying drugs directly to cells, so they can aid in treatment diagnosis and can also be formulated into antibody drug conjugates as a targeted therapy. And again, we will discuss more of these targeted therapy in the biotechnology series. Besides monoclonal antibodies, other notable biologics include cytokines, fusion proteins, enzymes, and growth factors, and these can all be made through recombinant DNA technologies. Now, here is a pie chart illustrating the breakdown of biological products by market sales. It highlights the dominance of monoclonal antibodies and the relative market shares of other biological product categories. Now, to conclude this topic, let's discuss some key challenges associated with biological products. First one being is anti-drug antibodies, commonly abbreviated as ADA. Now, one of the major hurdles is the development of anti-drug antibodies. These antibodies originate from the patient's immune system and are designed to neutralize the therapeutic monoclonal antibodies. Since many MABs have an animal origins, they contain foreign proteins which trigger an immune response, and our immune system is naturally programmed to eliminate foreign substances, leading to the gradual production of antibody that can anti-drug, also called the anti-drug antibodies. And there are several types. Type 1 ADA, now these antibodies directly bind to the antigen binding site of the therapeutic mAb, rendering it ineffective. Other ADAs, including some that do not bind directly to the active site but still interfere with the drug's function, making their effects less predictable. Now, they also poses detection challenges. To identify ADA is quite difficult because most exist in free-floating form in serum and vary significantly from patient to patient. And this lack of standardization makes ADA detection highly complex. Another key challenge is the presence of anti-PIG antibodies. Now here PIG stands for polyethylene glycol or pegylation, a process used to modify biologics including mRNA vaccines to reduce renal clearance and increase half-life. Pegylation can enhance drug activity by 5 to 100 fold. However, pegylation can also increase molecular size, making the drug appear foreign to the immune system. In some cases, the body mistakenly recognizes pegylated molecules as threats and produces anti-pig antibodies. The impact has a lot. Now, it can impact drug efficacy when anti-pig antibodies bind to the pegylated surface. The drug loses therapeutic effectiveness, and there are also clinical significance. Many biologics actually fail in late-stage clinical trials due to immune responses against pig. Now, that concludes our introduction to biological products. We will explore mRNA vaccine technologies and other similar technologies in more detail during our upcoming biotechnology lecture, and I'll see you later.